track the course of these alleles. I've tagged one as maternal or mat and one as paternal. So we know that the first thing that happens is they have to duplicate, right? And they don't duplicate like this, right? Because that's not correct. We know that they become homologous sister chromosomes. So because they're homologous, we get 2 copies of each color together. So 2 reds. I'll just draw the alleles again just to keep it simple. A, B. All right, cool. We tracked this so far? Yeah? All right, cool. Yeah. And then now, this is still in one cell, and now we can start dividing, right? So what's the first process in meiosis that gives us genetic variation? Yes?
Crossing over.
Yeah, crossing over, right? Now, crossing over can happen at any point, right? Usually, it's just donated or indicated as like happening in a particular corner, right? Any chromatid can swap and change material, but like diagrammatically, it's probably really confusing when I show this chromatid swapping with this one, right? Because then it's like going across several other ones, which is why in diagrams, you probably only ever see like these 2 that are closer together swapping or these 2. That's just because it's easier to represent and provides little confusion. Sometimes the middles can swap, but for this diagram, I'll just swap my bottom 2. I'll label it here. Do we remember what phase this process occurs in? Yeah?
Phase 1.
Yeah, prophase 1, right? Okay, so how do I draw this? This is what I get after crossing over, right? So again, like I said, it doesn't just have to always be this spot. Like technically, this blue one or this green one, whatever, it could have crossed over with this chromatid here. But the reason why, like it's possible, but the reason why you don't see it as much is because people can't be stuffed to make the diagram that complex, which is why we just swapped the 2 closest ones. Continue. So I realized that I only drew 2 pairs, which is not the best way to show independent assortment, but we'll just work with it, right? So let's say I have this. We know that independent assortment occurs in metaphase 1, and I guess the idea behind it is they can line up on either side of the middle and then get pulled apart, right? So what that means is if I'm just going to draw a cell in the middle, I have 2 options, right? So I'll just say this is option 1, and then this is option 2, right? So in option 1, I'll just have, let's just say that this one chooses to line up on the left side. Right, and then this one chooses to line up on the right side. Right, like this, right? So this is just random, right? Because this one's on the left, this one's right. Or it can happen the other way around, which means that in this case, this one has lined up on the right side now. Cool. Can we see the difference here? Yeah, right? So we know that they get pulled apart after this phase, right? They're still joined together at the chromatid, but they're going to get pulled apart, right? Which means we get like various resultant gametes. And obviously, it's probably better with another pair below it, but when they get pulled apart to different gametes, which means that your arrangement should be different. So this is the second way we get variation. Right now, one of the HSC questions, or what they love to ask for meiosis, is we might ask you for all the possible combinations of gametes that you can get from this diagram, right? We did a question on it a while ago, but I can just, I'll just model that for, let's just say option 1 over here, right? So we know that we should get 4 sets of gametes from meiosis, right? Because meiosis is reduction division from 1 to 4. We still have our other PMAT going on, but I'm just going to gloss over that, right, briefly. But we know that these would get separated, right? So this is metaphase 1, and anaphase 1, they'll be pulled away, right? And then telophase 1 will happen, and they'll be cut, right? So that means at the beginning of the second phase of meiosis, these two will be in their separate cells, right? 1 cell, 2 cell, and then they'll split again, right, for the final division. So I'll just draw what that will look like on the board for us, right? So, so in prophase 2, right, we'll have 2 cells, right? That should become 4 eventually, and I won't draw the alleles just to save time, but it'll be something a bit like this, right? It looks like this in prophase 2, right? And we know that they get pulled apart again in anaphase and telophase. So if I'm going to draw my 4 gametes, I should have 4 circles, right? Such a classic question, right? And then each of these circles will just contain one of these options, right? So this first one, these first 2, actually no, these 2 here, I'll do for this option. So that means my first allele is capital A and capital B. My second allele is capital A and lowercase b. My third allele is lowercase a and capital B. And my fourth allele is A and B. Right? Cool. So these are your gametes, right? Hopefully this makes sense. Right. But I think the important part of meiosis, and the reason why I went through it again, is just understanding exactly how crossing over occurs, right? At which point it happens. Not getting mixed up with the fact that when they kind of duplicate at the beginning, they're still like homologous sister chromatids. And then knowing what independent assortment is and how that can affect your final combinations when they split for the second division. Cool?